So, good morning to all of you. Today, I'm going to discuss with you all about the aneuploidy. In the last videos, we have discussed about the euploidy, that is the monoploidy and the polyploidy. So, let us discuss about aneuploidy today. Now, what does aneuploidy means? It simply means change in the number of chromosome from the chromosome sets. That means either you have a missing chromosome segment or a whole chromosome may be lost. Sometimes chromosomes, non-homologous chromosomes may also be lost from two different regions. Sometimes you can gain one extra or more than one chromosome. So aneuploidy simply refers to change in the chromosome number, not change in the chromosome number sets. So it simply means that change that involves parts of a chromosome set resulting in individual are called aneuploids. So the where your chromosome number is going to be changed from the set of chromosome is are called as aneuploids. Okay, and aneuploidy can either due to the loss of one or more chromosome known as hypoploidy, or due to addition of one or more chromosome to the complete chromosome set. So hypoploidy, as I have said in the first video of your monosomy, in your monoploidy part video, that hypoploidy means loss of one or more chromosomes, whereas addition of one or more chromosomes is known as hyperploidy. So hypoploidy mainly occurs due to subtraction. So due to subtraction or loss of a single chromosome, hypoploidy can occur. That is called as monosomy. Sometimes double monosomy can also occur, where from the diploid set of chromosome, two chromosomes from non-homologous regions has been lost. Sometimes a whole chromosome pair may be lost from the set of chromosome, that is called as nullisomy, that is called as 2n minus 2. So here it is written that loss of one pair of chromosome is called nullisomy. So if a pair of chromosome is lost, suppose we have 23 pairs of chromosome and one pair of chromosome is lost, suppose the chromosome number 21 has been lost, so we have now only 22 sets of chromosome. Okay, we have only 22 sets of chromosome, that is called as nullisomy. But in monosomy, it simply means just loss or subtraction or loss of a single chromosome, that is called as monosomy. Okay, so two things can be observed, monosomy and nullisomy, where the homologous chromosomes are being lost in nullisomy, but in monosomy the non-homologous chromosomes are lost. Likewise, hyperploidy means addition of one or more chromosomes, right? Hyperploidy may involve addition of either a single chromosome. If a single chromosome is being added to the chromosome set, it is called trisomy, which is known as 2n plus 1, or a pair of chromosome may be added, known as tetrasomy, 2n plus 2. Okay, so these are the concepts of aneuploidy, that a change in the number of chromosomes, either subtraction of the chromosomes or addition of chromosomes from within the chromosome sets. Let us discuss about the monosomy now. So it is saying in monosomy that your diploid organisms which are missing one chromosome of single pair are monosomic. So those diploid organism, that means we have two sets of chromosome uh, which are missing one chromosome of single pair. From that single pair of chromosome, one chromosome is being lost, that is called as monosomy, with the genomic formula 2n minus 1 that I have said. So in monosomy, it simply means the organism lack one chromosome. The general chromosome formula is 2n minus 1. Now sometimes if more than one chromosome from the other chromosome pair is lost, it is called as double monosomy. Okay. So again I repeat that if one or more chromosome of some other chromosome pair is being lost, then in an individual, then they are known as double monosomics, which is known as 2n minus 1 minus 1. That means this loss of one chromosome is from different regions, from different chromosome, and this is from the different chromosome location. Okay. Next, it is saying that the sorry. Next, it is saying that the n minus 1 gametes do not survive in plants, but in animals they may cause genetic imbalance. That's why different type of disorders we can see in monosomy. So n minus 1 gametes do not survive in plants, but in animals they may cause genetic imbalance, which is manifested by high mortality, that means death rate will occur more, reduced fertility, but that means they will be sterile, and of the resulting organism. So, so these n minus 1 gametes will not survive in plants, although we are trying to prepare that type of monosomic plants to study the gene character, study the alleles, the different versions of the same gene. But in animals, if monosomy occurs, it may cause genetic imbalance. It increases the mortality rate, the death rate of that individual, and it will also reduce the fertility of that resulting organism. Monosomics shows variable reduction in the vigor, means all the fruiting, all the growth of the plant, the healthiness of the plant will reduce size, fertility of the plant like maize, tomato, and wheat. Now it is saying that monosomy in case of diploids is not tolerated. Why? Why monosomy in diploids is not tolerated? Because it is creating imbalance due to loss of one complete chromosome. So monosomy in diploids is not tolerated since it creates imbalance in the chromosome constitution due to loss of one complete chromosomes. A complete chromosome is being lost. So that chromosome may have the alleles of dominant type that shows the dominant characteristics. So the other chromosome from that homologous pair of chromosome will now be expressed that may have some lethal, that may have some recessive characteristics. But those who are polyploids, monosomy has no apparent effect because they have several chromosomes of the same type, and loss of one chromosome can be easily tolerated. Now, those who are polyploids, like those who have 3 sets of chromosome or 4 sets of chromosome, even they lost one chromosome, that means if they have 3n minus 1, it will have no impact because they have that chromosome more. Okay, they have 2 more sets of chromosome in case of triploid. In case of tetraploid, they have more 3 sets of chromosome if one chromosome is lost, right? So in polyploids, monosomy has no such aberrant effect because they have several chromosomes of the same type, and loss of one chromosome can be tolerated. Okay. and it is saying that the number of possible monosomics in an organism will be equal to the haploid chromosome number. Here it is written, the number of possible monosomics in an organism will be equal to the haploid chromosome number. What is the possible number of monosomy that can occur in the organism? That is equal to the haploid chromosome number of that particular organism cell. Like for example, common wheat have 21 pair of chromosomes, so their haploid number is 21. So there are 21 possible monosomics that can be known. So in common wheat, since they have 21 pairs of chromosomes, so their haploid chromosome number is 21. So 21 possible monosomics can be possible, right? These 21 monosomics in wheat are produced by ear shears in a variety called Chinese spring. And this type of monosomy is used in for studying the genetics, for genetic study, for studying the alleles in that particular of that particular deleted chromosomes. Okay, and that they are known as Chinese spring. This type of variety that has been created by E.R. Shears, known as Chinese spring, and being used for genetic studies all over the world. Similarly, there are also double monosomics and triple monosomics as well. Double monosomics means if two chromosomes are lost, that means 2n minus 1 loss of a chromosome minus 1 loss of another homologous non-homologous chromosome. So if one or more chromosome of some other chromosomal pair, if one or more chromosome of some other chromosomal pair is lost in an individual, this is known as double monosomics. Okay. Similarly, triple monosomics can also be occur where three chromosomes are being lost from the chromosome set. That is called as triple monosomics. Okay. So since monosomic lack one complete chromosome, they are genetically imbalanced, and so they are either lethal, and lethal means they are deadly. There is a possibility of high mortality, high death rate, or they have reduced viability. Sometimes partial monosomy can also occur where the individual does not have a portion of the chromosome while the rest of the chromosome have two copies. Like for instance, we have short arm and long arm, right? We have short arm and long arm. This is a centromere. The short arm is known as the P arm, whereas the long arm is known as the Q arm. So suppose the P segment suppose a short segment of the P arm is lost, that can also cause, that is also known as partial monosomy. It is known as partial monosomy. Okay. Now let us see about monosomy can be easily be produced in polyploids. This has not been said, but it has been said here in this that in polyploids monosomy has no apparent effect because they have several chromosomes of the same type, and loss of one chromosome can be easily tolerated. So we can create monosomy in your polyploids, which can be useful for our genetic studies, right? So monosomies can be easily produced in polyploids because they possess more than two chromosomes of the each type, and loss of one or more chromosome could be easily tolerated. And I have already said that the number of possible monosomics is equal to the half-life chromosome number. Now let us see about the human conditions due to monosomy. The first one is your Turner syndrome. Now in humans, this Turner syndrome is the only full monosomy that can be observed, where the X chromosome, we have 23 pairs of chromosome, we know. Out of this 23 pair of chromosome, 22 pairs are known as the autosomes. Autosomes. And the last pair, the 23rd pair, is the sex chromosome pair. So within that sex chromosome pair in females, they have two X chromosomes, and one of the X chromosome has been removed out. Um, that is Turner syndrome actually. So in humans, Turner syndrome is the only full monosomy that can be observed. The rest of the full monosomy occurs among the individuals are lethal and cannot able to survive during developmental phase. So we can only see this Turner syndrome as full monosomy. Other full monosomy can be possible, but they are deadly. They will not develop, they will die before developmental stage, okay, during developmental phase. So what happens in Turner syndrome? You have studied in your class 12 examination. It has been given in your syllabus that people with Turner syndrome typically have one X chromosome instead of the two X chromosome. Instead of having two X chromosome, the female have only one X chromosome. So the sex chromosome pair is getting deleted. One of the sex chromosome is going to be eliminated. Now this type of syndrome mainly occurs among females. Affected females will have this type of feature: sterile, that means they cannot able to produce the ovum; short in stature, short in height; low set of ears and broad chest. Low set of ears means the ears set will be lower, and broad chest. And it has been shown that in one in every 25,000 birds will exhibit Turner's syndrome. Second type of syndrome is Cry-do-chat syndrome. This you have also studied. It is also known as 5p deletion syndrome. It simply means that the fifth chromosome, their short arm, which is the P arm, somewhere in the region of the P arm of the fifth chromosome has been eliminated, has been deleted, and that leads to the formation of Cry-do-chat syndrome. Now it is caused by deletion in the short arm of chromosome number 5, which is known as partial monosomy, and thus during infancy stage their crying of the infant will comparable to the crying of like a cat, mewing of a cat. And due to malformation of the larynx, your voice box is known as larynx, that larynx get malformed, means abnormally formed, your nervous system gets abnormally formed, you will find difficulties in communication among the other individuals. It will be difficult to communicate with the other individual when you are suffering from this syndrome. And obviously the IQ levels are also being lower because mental retardation will occur. Third type of example of your monosomy is IP36 deletion syndrome. IP36 deletion syndrome, I not IP36, 1P, it is 1P, 1P36 deletion syndrome. 1P refers to the 1 refers to the chromosome number 1, P refers to the short arm, and 36 is the position in the short arm which is going to be deleted. That leads to the formation of this type of syndrome. So it is also a partial monosomy because only the 36th position of the short arm of chromosome number 1 is going to be deleted at the end of the short P arm of the chromosome number 1. What are the symptoms include in this IP36 syndrome? Delayed growth, seizures, hearing and vision impairment. There will be problem in the hearing and vision in your looking. distinct facial features. Your facial features will be different from the normal person. The face shape. Microcephaly, this term means small head, low muscle tone. The muscle arrangement will be peculiar, and this deletion syndrome is estimated to occur approximately 1 in 10,000 live births, and is most frequently occur um, in telomeric deletion. Telomeric deletion means the ends of the chromosome are called the telomeres. That regions are mostly deleted. The chromosome number 1 telomere, if this is the chromosome number 1, its telomeric regions are on the end sides. These regions are going to be deleted. That leads to formation of this IP36 deletion syndrome. Okay. So this was about the monosomy. Now let us discuss about the nullisomy now. Okay. What is nullisomy? What is nullisomy? It is saying that the cell, tissue, or organism in which both the homologous of a chromosome are being missing from the somatic chromosome complement is called nullisomy. So when a cell, tissue, or organism, within that cell, tissue, and organism, homologous pair of chromosome are being lost, are missing from the chromosome set, is called nullisomic. That's why the genomic formula is 2n minus 2, and this condition is called as nullisomy. Suppose we have 23 pairs of chromosome, and out of this 23 pair, the 22nd pair of chromosome is being lost. That is called as nullisomy. For example, I have said. Now, if two pairs of homologous chromosome are being absent, suppose if two pairs of homologous chromosomes are absent, then that cell, tissue, or organism will be called as double nullisomic. That means we have 23 pairs of chromosome, and out of these 23 pairs, our second pair of chromosome and the fifth pair of chromosome are being lost. That will be called as double monosomic. For example, I have said nullisomic, obviously they will not survive in diploids, but they can survive in polyploid species, because polyploids have extra sets of chromosome. They have extra sets of chromosome. So even if a pair of homologous chromosome from that set is being lost, they have other two pairs as well. But those who are diploid, like normal human beings, they, if a chromosome pair is being lost, if a chromosome pair is being lost, it will be a lethal. Okay, it will be lethal, deadly, which will not able to survive. Nullisomics can be obtained in self-progeny of monosomics, where n minus 1 type male and female gamete fuse to form 2n minus 2 zygote. So you can create, we can able to prepare nullisomics in the self-progeny of monosomics, where n minus 1 type male and female gametes, they fuse to form 2n minus 2 zygote. So the gametes, male and female gametes having the n minus 1 type, that means they have they have missing one chromosome. They same chromosome, they fuse to form nullisomic zygote. So the males having n minus 1 in within their chromosome, that one chromosome is missing, and the females, they have also one chromosome missing, same chromosome missing as in case of male. When they are fused to form zygote, we get a nullisomic individual, nullisomic zygote. Now through nullisomic analysis, why we do nullisomy, why we create nullisomy in polyploids, it is used to determine the genetic linkage actually. to find out the linkage between the two genes, how closely the two genes are linked together, how closely they are placed within the chromosomes so that they can inherit it together during segregation of the chromosome to the opposite poles during your meiosis, during your mitosis. And in this way we can study the traits, we can study the characteristics that are controlled by these groups of genes. And this method can also be applied to tissue culture as well, plant tissue culture uh, of human not it is applicable to tissue culture in plants also as well as in animals also, to studies the human nullisomic cells. So here again I have said about the nullisomy. So nullisomy is an organism which has lost a chromosome pair. So an organism who has lost a chromosome pair is called nullisomic, and nullisomic organism has the genomic formula of 2n minus 2. A nullisomic diploid do not survive, but a nullisomic polyploid can survive, but it will reduce its vigor, its growth will be affected, and its fertility rate will also be affected. Like if we can able to induce nullisomy in hexaploid wheat, it will still grow, but their vigority, their fertility will be much lower. Next is your trisomy. Trisomy or trisomics. Let us see. Now, trisomy or trisomics are those diploid organisms which have an extra chromosome. So here it falls under the category of hyperploidy, where extra chromosome is present. If one extra chromosome is present, it is called as trisomy. An organism who have this extra chromosome are called as trisomics. Since the extra chromosome may belong to any one of the different chromosomes of the haploid complement, the number of possible trisomics will be equal to the haploid chromosome number, just like monosomy. That in trisomy, the extra chromosome belongs to any one of the different chromosomes of that haploid complement. That's why number of trisomics will be equal to the haploid chromosome number. Haploid chromosome number. Okay. Now, trisomy can be of many types like primary trisomy, secondary trisomy, tertiary trisomy. Okay, what is primary trisomy? If I say, I will not go into a diagram now because it will confuse you. Okay, it is saying, okay, so it is saying that when an extra chromosome is identical to its homologs, such type of trisomy is called as primary trisomic. So when that extra chromosome that we are having 2n plus 1, that this extra chromosome is identical to one of its homologs within the chromosome set, that is called as primary trisomic. So let us see the primary trisomy diagram. Okay, so this is the primary trisomy. You can see 1 2 1 2 1 2. These are the regions, similar regions. So three chromosomes. This set is identical to this homologous pair. That's why it is primary trisomy. What does secondary trisomy means? In secondary trisomy, what does it says that in secondary trisomy it means that the extra chromosome should be an isochromosome. It simply means that both chromosome arms are genetically similar. We have a P arm and Q arm, the short arm and long arm. If the chromosome has an identical copies of that chromosome arms, and if it is getting to be added to the chromosome set, that is called as tertiary, that is called said to be as secondary trisomy. Let us see with the help of diagram. You can see here we have a three sets of chromosome, we get three sets of chromosome, but the third chromosome, the extra set segment of chromosome, they have two identical arms which are identical to a portion, to the portion of the homologous chromosome. Now this type of chromosome is called as isochromosome. Isochromosome. Why? Because they have identical arms. Now we have named them as 1 2 1 2 1 2. Okay, but actually that is p arm and q arm. Suppose the two p arms are similar of this chromosome, that type of trisomy is called secondary trisomy. Okay, where identical arms of a chromosome is being added to the chromosome set. And the last is the tertiary trisomy. In tertiary trisomy, what happened? In tertiary trisomy, it is saying that a tertiary trisomy would mean that the extra chromosome should be the product of translocation. Translocation simply means where the segment of a chromosome has been shuffled with the others, with the segment of other chromosome. Let us see with the help of example. You can see here there are two sets of homologous chromosome 1, 2 and 3, 4. Now this is an extra set of chromosome which has been added, but it has the portion of this homologous chromosome and has the portion of this set of homologous chromosome. So it has been translocated. That means it somehow gets the segment 1 and the fourth segment has been translocated and form this type of translocated chromosome and being added to this region. Okay, now such type of thing occurred due to non-disjunction, due to not proper crossing over. Okay, uh, due to crossing over at unexpected regions, non-homologous regions. Okay, mismatches, non-disjunction, not proper segregation of the chromosomes to the opposite poles leads to the formation of this type of translocated chromosome. Sometimes external factors also play an important role, like UV rays, like your chemicals. Some mutagenic agents are there which cause mutation, which cause change in the base pair, cause deletion of a segment, and that deleted segment goes and joins to segment of a non-homologous chromosome, that is called as translocation. So due to which this type of event, tertiary trisomy, can occur. Now let us see the examples of trisomy. Down syndrome. Down syndrome means trisomy 21. Trisomy trisomy 21, that means the 21st chromosome, there are three copies of your 21st chromosome. That's why the name comes trisomy 21. So in Down syndrome, it is named after the physician John Langdon Down, who first described this genetic defect in 1866, and it is also known as Mongolism. And this is occurring in human autosomes because the 21st chromosome in us is actually an autosome. The first 22 pairs of chromosomes are autosomes, while the last pair, the 23rd pair of chromosome, is called the sex chromosome. So it is occurring in the autosomes, so it is functionally important, right? And it is referred to as trisomy 21 or 47 XX or XY21. Now, this Down syndrome is most common chromosomal abnormality in live births. Out of 650 live births, one can suffer from Down syndrome. There are about 50 physical characteristics shown by the Down syndrome infant soon after birth. Soon after birth, the infant shows some characteristics which signifies the Down syndrome. Like, what are the characteristics? Mild or moderate mental retardation, IQ level is low, eyes that are slant up with internal epicanthal fold, tongue which will be large and swollen, and protruding, means large tongue, underdeveloped ears, short stature, stubby fingers, the fingers are not developed, enlarged liver and spleen. Down syndrome children will have congenital heart defect, and they will be affected by, they can have a risk of affecting of acute leukemia, and they have a broad skull, hyperflexibility of joints. It has been observed that women who are 25 years of age, they are more likely to give birth to a child having Down syndrome compared to the women who are aged as 20. So it's a very critical thing to conceive child um, at 45 years of age, because the child is having more risk of having affected with Down syndrome, this type of genetic disorder. And the main cause of Down syndrome is your non-disjunction, not proper separation of the chromosome number 21 during oogenesis. You know about oogenesis, development of the oocyte from the primary oogenia, which is the main cause of occurrence of trisomy 21. So if the oogenesis does not occur properly, it leads to this type of trisomy 21. Okay, so this is the thing, this is the example of your trisomy. Other examples are also there. The next syndrome that I am going to study is about Edwards syndrome. Edwards syndrome is also known as trisomy 18, 47XX or XY. It is first described in 1960 by John H. Edwards and his colleagues. Trisomy 18 is found to contain an incidence of 0.3 per thousand births. So it has more rarely incidence that out of 10,000 births, out of 1,000 births, 0.3 people will suffer from Edwards syndrome, and um, it is characterized by multiple malformations. That means your normal formation will not occur. Primarily low set ears, small receding lower jaw. The lower jaw will be receding, clenched fingers, flexed and clenched fingers. Your heart muscle will be malformed, cardiac malformations, various deformities of your skull, face, and feet. The hair lip and the cleft palate, which is in Bengali we say talu. Okay, it often occurs. It often occurs. It may get removed. Okay, death take place around 3 to 4 months of age. This is the main thing that we can see. Trisomy 18 children shows evidence of severe mental retardation. They lost their intellectual ability, which is more pronounced in female. It has not been clear that why female who are suffering from trisomy 18 have severe mental retardation compared to male. It has not been clear. Like Down syndrome, occurrence of Edwards syndrome is too related with maternal age. Now, if the maternal age is 35 to 45, older mothers, if they are going to have a child, then the possibilities of having Edwards syndromes is little bit more increase. Third is your Patau syndrome. Patau syndrome is your trisomy 13. Um, that means your 13th chromosome has been occurred 3 times. This syndrome was described in 1960 by Klaus Patau and his co-workers, and it occurs 1 out of 20,000 live births. Individual having with Patau syndromes, they are mentally retarded. All are almost the same thing: sloping forehead, hair leaf, cleft lip, and cleft palate. So their cleft palate is being not formed. As you can see in the figure, the cleft palate has not been formed. That leads to this type of syndrome. Polydactyly, that means you have an extra finger, okay, is of more always present. The hand and feet are deformed. Cardiac and various internal defects, kidney, colon, small intestine, there will be defect in your this internal body organs, and are common. Death usually occurs within hours and days. Death usually occurs within hours or days, but the fetus may abort spontaneously. Sex chromosomal variations will be, which has been shown in case of Patau syndrome. So these are the actually the images that has been shown. It has been shown that how the trisomy chromosomes arrange themselves during metaphase. So if they have a primary trisomy, the extra chromosome that you can see, this extra chromosome is look alike like the normal homologous pair of chromosome, then they will arrange like this in metaphase 1 of meiosis. Now if there is secondary trisomy where the extra chromosome is an isochromosome, you can see the extra chromosome is an isochromosome having similar segments, then they will arrange like this in metaphase 1 of meiosis. And finally, if the trisomy that is going to be occurred is tertiary trisomy, that means the extra chromosome that is being occurred is an translocated chromosome, then they are going to be arranged like this, which is a pentavalent like structure, looks like this in your microscope. Okay, more like dumbbell shape. Last is your tetrasomy. Tetrasomy is the diploid organism having two extra chromosome are called as tetrasomic. Genomic formula is 2n minus 2. That means two extra chromosome are being now present. That leads to tetrasomy. All the 21 possible tetrasomies are available in wheat. So, what is saying about tetrasomy? That your tetrasomy is a form of aneuploidy with the presence of 4 copies of chromosome instead of normal 2 copies of a particular chromosome. The main cause of tetrasomy, as we know, non-disjunction, that is failure of the homologous chromosomes or sister chromatids to separate properly during cell division. I am zooming here. So non-disjunction similarly means failure of the homologous chromosome or sister chromatids to separate properly during cell division is known as non-disjunction. So the main cause of tetrasomy is due to non-disjunction in meiosis, where the cells are dividing during meiosis 1 stage, during meiosis 2 stage, for the formation of gametes, for the formation of egg and sperm cells. So due to non-disjunction, tetrasomy occurs. As a result, that extra chromosome is found in the sex cell, it is found in the sperm or in the egg, and after fertilization, the resulting fetus will have extra chromosome instead of typical normal chromosomes. Like it will have 48 chromosomes instead of having 46. There are various examples of tetrasomy like tetrasomy 18p. Tetrasomy 18p means the 18th number of chromosome, the short arm of the 18th chromosome. Short arm is the P arm. This has been 4 times it has been found. So tetrasomy 18p is a very rare chromosomal disorder in which the short arm, the P arm of the 18 chromosome appears to be 4 times rather than twice in the cells of the body. Now sorry, individuals with normal chromosome makeup have two 18 chromosome, both of which have a short arm and a long arm. But here, in case of tetrasomy 18p, the individuals will have the short arm of the 18 chromosome 4 times instead of having twice. Now, people who are suffering from tetrasomy 18p will have changes in their muscle tone. What is muscle tone? Muscle tone is the continuous, passive, partial contraction of the muscle that helps to maintain the postures when you are sitting, when you are running, when you are walking. All those postures are maintained by your muscle. So you will have a you will have a different type of muscle tone if you suffer from tetrasomy 18p. This may have increased or decreased muscle tone. So we can have an increased muscle tone known as hypertonia, or decreased muscle tone called hypotonia. Hypertonia means a condition in which there is too much muscle tone, so that the arms or legs, for example, become very stiff and are very difficult to move. And muscle tone is regulated by some signals that travels from brain to the nerve cells and tells the muscle to contract. That is going to be inhibited, not properly function. Decreased muscle tone is called hypotonia, where the decreased muscle tone uh, it extends to a much flexibility to that affected individual when they lift their heads or their limbs. So this type of thing can occur who are affected with tetrasomy 18p, and it also contribute to delayed development of your motor skills. Motor skills are those movements or action that are involved in muscles, like um, sitting, crawling, walking. These are the motor skills actually, which will be delayed. Those who are suffering from this 18p deletion 18p syndrome. Babies with tetrasomy 18p will have trouble in feeding and may vomit frequently, which makes it difficult to gain weight also. Additional features of tetrasomy 18p are jaundice, seizures. Seizure is a disorder in which the nerve cell activity in the brain is disturbed. Okay, vision problems will occur, constipation will occur throughout their lifetime, and cryptocorchidism occur in case of male. Cryptocorchidism, as you have studied in your class 12 reproduction chapter, that it is the condition where the testis has not comes out in the scrotal sac in male. So scrotal sac are the parts of the male reproductive organ where the testes are being located to maintain the formation of sperms in a certain particular temperature. But in cryptocorchidism, the testicles has not comes out from the abdomen. It has not comes into the inside into the scrotal sac. It remains in the body of the abdomen of that human being. So this type of cryptocorchidism may occur. Other examples are also there, like tetrasomy 9p, Pallister-Killian syndrome, cat's eye syndrome. This you study. Don't go for thoroughly. You go thoroughly, but not too much in detail. The important points has to be said. Just go through it. You can able to start. We can able to easily learn these things. Okay. So this is the Pallister-Killian syndrome. I am giving these notes to your WhatsApp group. Klinefelter syndrome, you always know, extra segment of X chromosome, extra X chromosome in male. You have studied in your class 12 syllabus. You just go through it. Means where what it occurs and what effect it occurs, symptoms, best. That's much. The last is your non-disjunction. I have studied about lots that all this aneuploidy occurs due to non-disjunction. What is non-disjunction? First, let me discuss about disjunction. Disjunction is the another word for normal segregation of homologous chromosomes or chromatids to the opposite poles at meiosis or at meiotic division. But non-disjunction is the failure of this process, and two chromosomes or chromatids may go to the one pole and not to the other. In simple diagram, if I say, suppose this is the cell undergoing a cell division stage, and this is your mitotic spindle. This is your mitotic spindle, and in this mitotic spindle the chromosomes are being attached. The chromosomes are being attached. Now they will migrate to the opposite poles, and they will migrate to the opposite poles. But if suppose spindle is going to be disrupted, then what happened? Suppose look at this image, look at this portion. I have removed this spindle here. So this spindle, this one, it will take that whole chromosome set to the opposite poles, and the other the other daughter cell will not get that chromosome at all. So during gamete formation, especially in meiosis, as they don't get that chromosome, it leads to loss of a chromosome segment, that is your monosomy, whereas the daughter cell or the gamete which is receiving that extra chromosome leads to trisomy. So this type of event leads to non-disjunction. Okay, failure of the chromosomes to migrate to the opposite poles. So non-disjunction is a failure of this process. The two chromosomes or chromatids or chromosomes go to one pole and not to the other. In meiotic non-disjunction, the chromosomes may fail to separate either at first meiotic division or at second meiotic division. Either way, n plus 1 gametes or n plus 2 gametes will be formed. Now, this type of non-disjunction can also occur in mitosis as well, but it is not too much of lethal, because in mitosis what happened, it is distributed among the daughter cells, but it is not commonly occurring in all somatic cells. Okay, because mitosis occur in all somatic cells, we know. So it is not so much lethal, it is not hereditary. But in meiosis, if this type of non-disjunction occurs, then it is a hereditary problem. Okay, because it is actually distributed among the gametes, and the gametes will have either loss of a chromosome or gain of a chromosome part. Okay, and then when they are going to be fused with another haploid gamete like ovum, okay, or sperm, whichever may be, they form a zygote. That zygote will have that extra chromosome segment or loss of a chromosome segment. That becomes a hereditary. So meiosis, non-disjunction in meiosis is bit of problem. So again I repeat, again I am drawing the diagram for you to understand. Suppose this is your cell, and this is your this is your chromosome. First I'm drawing the chromosomes now in blue. Okay, I'm trying to draw the pair of chromosomes. Okay, so these are the pairs of chromosome. Okay, and these are the mitotic spindle which are drawn in green. Just three I'm drawing. We all know we had 24, 23 pairs of chromosomes, but for simplicity, I'm drawing this one. Now, this mitotic spindle is made up of microtubules, as you have learned in your cell biology class, and they help these chromosomes to migrate to the opposite poles, right? They help the chromosomes to migrate to the opposite poles so that they can be easily distributed among the gametes. But what if the mitotic spindle get disrupted? That means, suppose this is going to be eliminated, so what will happen? So as it is going to be eliminated, then what will happen? If you see it clearly, this portion, this one, as the whole chromosome has been, the two pairs of sister chromatids have been joined by this mitotic spindle, so it will migrate to this pole and the gamete is receiving that extra chromosome, while this, as the mitotic spindle has not been formed here, it is receiving that, it is losing that extra chromosome, so it will be -1, this will be +1. So in this way your non-disjunction occurs. Okay, so it is saying that non-disjunction, sorry for that, non-disjunction occurs spontaneously and can be increased by interference with the microtubule action. So if you can interfere the working of microtubule, you can spontaneously, you can create non-disjunction, and it occurs more frequently at meiosis 1 than that of meiosis 2, as normal anaphase 1 disjunction required that proper homologous arrangement of the chromosomes to be maintained at prophase 1 and metaphase 1. Okay, so the same thing I have drawn here, you can see. So this is the non-disjunction at first division, where you can see the whole chromosomes are migrating to one pole only due to failure of the microtubule association, and that's why in the second meiotic division, when they form the gamete, they get an extra chromosome while the other gamete is not getting that chromosome, so it leads to n-1. Here I have shown only the one chromosome pair. There are 23 pairs of chromosome, remember. This event is occurring only on one chromosome pair. Okay, similarly, so this type of event leads to aneuploidy like condition, the n+1 and n-1 like condition. But non-disjunction can also occur in the second meiotic division as well, if you see. So the first division appears normal, okay, but the second is in the second division non-disjunction occur, which leads to failure of separation of the chromosomes to the opposite poles, and that's why aneuploid like condition occurs in some of the cases partially. So this was all about your aneuploidy. I hope you have understood this part, especially the non-disjunction. I will try to give more notes about the non-disjunction in your WhatsApp group. In the next video, I will try to discuss about the structural changes in chromosome, that is your deletion, duplication, inversion, and translocation. And that's all for today.